Welcome, everyone. This is the second session of the spring series. As you know, we're focusing on aging, understanding the mechanisms underlying epigenetic changes in aging. And today we're welcoming Ken Raj from the Altos labs at Cambridge Institute of Science. Stan will be introducing Ken. So it gives me pleasure to introduce to you Stan Botchway. I think many of you know Stan has been a long-time collaborator with the foundation and is one of our scientific advisors. And Stan's laboratory is at the Central Laser Facility at Harwell under the STFC UKRI banner. And so Stan is going to give a short talk and then a more formal introduction to our speaker. So without further ado, Stan, if you'd like to share your slides and kick off.
Thank you very much, Jeffrey. Last webinar, Pedro gave us an excellent talk on the role of genes in aging. But I just want to take these few minutes to do a bit of a plugging for proteins. I know Ken is also going to talk about epigenetics, but I just thought proteins are perhaps one of these dark horses which we tend to forget in their roles in biological functions. And I've been working with proteins for quite some time. And initially, I was looking at how proteins repair DNA, trying to image protein function in real time and down to the picosecond timescales. And what you tend to find is the function of proteins are actually not quite what you think. So I just want to take one other protein, which is where I actually met Ken. Oh, if I can get my slides to move. Right, so there we go. So mTOR, I came across mTOR about 15 years ago. This is when it all began, and mTOR stands for mammalian target of rapamycin. It's involved in so many different cell functions: cell growth, protein synthesis, nucleotide synthesis, immunity. Pretty much any function in cells and in the body, mTOR is involved in. And it was actually suggested to be involved in aging as well. And of course, there are people out there currently taking rapamycin, which is the main inhibitor of mTOR. Of course, it has its side effects. But the question is, how does mTOR actually function in aging? So what we tried to do in my team was to look at the complex of mTOR itself. So mTOR exists in two complexes. Complex one, can I get a pointer? If I get a pointer, no, I can't find a pointer. Anyway, so we've got complex one, which has a whole set of other proteins involved with the mTOR itself, and complex two. Both of these complexes function slightly differently. The question we ask is, how does the complex actually work together? And in the literature at the time, it was supposedly meant to be that rapamycin broke the complex apart. But we reasoned that there is no way the protein will fall apart and come together again when it's needed. It's too big. So there must be a different way in which it works. So that was our aim, to try and figure out how this protein works. And we also noticed by tagging the mTOR with fluorescent proteins such as GFP, mCherry, we can actually see the mTOR moving within cells. And we publish the mTOR C1 actually moving from the nucleus into the cytoplasm and vice versa. So it's actually doing something within the cell nucleus, and it is supposedly involved in DNA repair as well. But how does this work? We, through our fluorescence lifetime imaging, where we can see movement on the nanometer scale and picosecond timescale, we came out with the complex, how it might work. And what we did find is that the protein doesn't actually fall apart when you add inhibitors such as rapamycin. It only moves by a few nanometers, which actually makes sense, means that the protein just moves very slightly to function. And how does this therefore link into aging? This is where I think is very important, in the sense that if the protein doesn't fall apart and it only have to move very slightly, then you might have perhaps tunneling involved, and one then have to start considering quantum mechanical processes which might be taking place. So I just want to take this opportunity to make a bit of a plug for the complexity of proteins and how they might be perhaps where we should be looking at when considering quantum biology and aging. And in fact, this is where I met Ken. He was actually working on this MLS8 protein at the time, and also doing some very interesting epigenetic research. And I went to one of Ken's talk where he showed so many different things that are linked to aging, and simple things like your education can even determine your aging process, which I thought was just incredible. Anyway, moving on. I've known Ken for 8 years now, or perhaps more. Ken is currently PI at the Altos Labs Cambridge Institute of Science. Prior to his current role, he was head of the cellular biology group in the radiation effects department at PHE, what used to be Public Health England. Ken graduated with a biochemistry degree, followed by a PhD research in cancer virology at Cambridge University. I guess he's gone back to his roots where he is now. He then moved to the Swiss Institute for Experimental Cancer Research, where he conducted postdoctoral research into tumor viruses and cellular responses to DNA damage. After 8 years, I guess he had enough, he returned to the UK and joined the MRC, the Medical Research Council, at the National Institute for Medical Research, now the Crick Institute, to head a research group investigating replication of tumor viruses, the mechanism of action of anti-tumor viruses, and cellular DNA damage response. After this is when he then joined the PHE, now UK HSA, to further his investigation into cellular DNA damage and response. And here is where our paths crossed. He expanded his research into the mechanism of aging and the effect of low dose effects on histones in cell nucleus. He also had a particular interest in epigenetics and aging, and I guess that's what his talk is going to be about today. His title is, for his talk today, is Understanding the Mechanism, Understanding Epigenetics Changes with Age. Without further ado, Ken, I'll hand over to you.
So thank you very much for inviting me to this, to talk to you about epigenetics of aging. I'm in some ways glad that you had Pedro giving you the first one, because that makes life easy for me, because Pedro, I think, would have given you a fantastic primer into understanding the current concepts of how people are thinking about aging. Now, it is, of course, not a new topic. It's perhaps one of the oldest topic in research. Let me just see if I can move my... Right. Yeah. I mean, it goes back, if you think about it, it's not surprising that aging is a question that has been preoccupying us for a long, long time. Now, if you take a perspective of, let's say, people like, the folks like Plato and Aristotle, they saw aging as a disease. Now, that's quite profound if you think about it. If you think that aging is a disease, then you might think, well, maybe you can cure it if it is a disease. But that's not the only view, because then you get the view of Galen, all right, the greatest doctor ever, according to Galen himself. And, and he thinks that aging is not a disease. It's natural. It just happens. And if that's the case, again, it's profound, because then why bother trying to do anything about it? Because, it's just inevitable, like trying to do something about gravity. So those are the two, even though they're very old, and since then there were many, many hypotheses and models that were proffered to explain aging. They all cluster invariably into these two, either aging as a disease or aging as natural. And you might say, I've not heard of it in this way. Well, they do come about in other forms of description, such as aging is because of damage, most often compared to, a car wearing out. That's one. Or you can say, and some will say, no, aging is programmed into us, right? We are programmed to age. So those two those two are the modern equivalent of Plato and Aristotle's perspective and Galen's perspective. Now, I'm not going to go any further with that. I just want you to think about where do you think it is? Where is your opinion? Where does it lie? Now, having heard Pedro last week, you probably would have already got your perspective lined up there. And we'll see at the end of this talk whether you still hold the same perspective or, you might have changed your views on that. Okay, so just a very quick primer. Like I said, this talk is just going to be about half an hour. I don't want to overload anyone with this. It's just a very quick primer to get a few terminology right. Now, we are all very familiar with genetics. We know about DNA, ACGT, and things like that. And that's probably not going to be referred to very often in my talk. But what I want to talk about is epigenetics, which is what you see here. The CH3 that you see here, these are the methyl groups that are added to DNA. So those are the modifications that I'm interested in, and those are the modifications that we're talking about. And because and it's important to note that these modifications on DNA does not, they do not change the sequence of the DNA, okay? ACGT remain as ACGT, they are modified, and the site and the place or the nucleotide that I'm particularly interested in are only the Cs. Okay, so and this is what we refer to as epigenetics, above that of genetics. It doesn't touch the sequence of the DNA. Now, the C's that I'm going to refer to in my talk, I will either use the word cytosine, or when I do that, what I mean is cytosines next to G, guanine, which is CpG. So you can hear me say sometimes, you will hear me say cytosine or cytosins or CpGs, okay? They're the same thing. And when I say methylated cytosins or methylated CpGs, it means that particular CpG or CpG is modified by a methyl group. Okay, so that's the very quick primer to make sense of the talk if you're not familiar with epigenetics. Now, why epigenetics? Why do we think it is interesting and important? Now, we are all born with a set of chromosomes, and notwithstanding the fact that we do accrue a lot of mutations, meaning changes to our sequences, changes to our genetics, we acquire this because of, what we face with the environment, what we eat, what we do to ourselves. We can change all that. But genetic changes, which we refer to as mutations, are random, okay? As far as we know, we have not seen a mutation that is specific to a particular site, to a particular gene. It's random. You and I, we accrue mutations all the time, and they are random. To think that random mutations would lead to aging is possible, but it is a long shot, because aging appears to be quite a not straightforward, but quite a predictable process. You can look at someone and know roughly what their age is. Why? If it's random, how would you account for such predictiveness even by looking at them? Now, notwithstanding this improbability, there's still a lot of ideas about mutations leading to aging. Now, that's about as much as I would want to say about genetics and the mutations. What I want to do now is talk about what about that DNA methylation and modifications on the DNA that I've mentioned to you earlier. Now, when we are born, some of our DNA, some of our cytosins, okay, are methylated and some are not. And in function of age, okay, what you find is some of the cytosins now, let's say if we didn't have cytosins in some places, if we didn't have methylation in some places, in function of age, you will gain methylation. And places where you started off with methylation, you can lose methylation. And some places where you just remain unchanged. Now, this happens in everyone. The difference between epigenetics and genetics in this case is that while genetics is random, mutations happen in random places. These changes that we see in epigenetics, they can change in very specific ways and in very, very uniform way in every single person. So there are some positions in our genome where the cytosines would change in you and in me. Okay, so that's quite a remarkable thing to see in biology, to see something that changes so systematically and so uniformly. Now, to put it in an easier way for you to imagine, there are 28 million cytosins in our 3.2 billion nucleotide. If we just see this as a depiction of our genome on the left here, 3.2 billion nucleotides, and there are about 28 million cytosins depicted by each black line, let's say. Now, if you were to profile these cytosins, meaning if you were to measure each of these cytosins and see whether they are methylated at different ages, okay? You just take people of different ages and look at each position of the cytosins and measure and see whether they are methylated or not across different organs. What you will find is there will be thousands of cytosins that change with age in every single person and in all the tissues. Okay, that is extremely regular. It is so regular that about 10 years, as you can see over here, sorry, before I forget, it's very important to realize that when I say change, I mean like the red lines will be those that increase methylation, some of them with age, and some will decrease methylation with age in each person, okay, uniformly. And like I said, it is so regular, it is like clockwork. As such, about 10 years ago, Steve Horvath, who is in an office next to me, he used machine learning, subjected these changes to the machine, and what you could get out of it, a selection of just a few hundred of cytosins that you can take the values of these cytosins and methylation, and you can predict the age of a cellular population or a person just by looking at the profiles of the cytosins of these several hundred cytosins. Specifically, if you want to know how many, it's 353. We made you could do this with quite a few different variations, but you just need a few hundred cytosins, and you can tell the age of a cell, you can tell the age of a person, okay, from any tissue. Okay, just by measuring by measuring the extent of methylation of these few hundred cytosins. But I just want to stress the point here. These few hundred cytosins, they are representative of the thousands that change with age. Okay. And at this point, I would like to say there's nothing special about these few hundred. They are just the ones that are most predictive of age. It doesn't mean that they have got more biological meaning than the few thousands from of which they represent. Okay. Now, this algorithm that finally was Steve Horvath generated is referred to as the epigenetic clock, because we can use the epigenetics of a person and predict their age. And the age that we predict with this, we refer to as epigenetic age. So you will hear me say epigenetic age or DNA methylation age. They are the same thing. We use the words interchangeably. And they're extremely predictive because you can see over here, the graph here, these are samples of people of chronological ages from 0 to 90. And this on the x-axis is the predicted age of these people from their DNA. And the correlation is 0.96. It's a remarkably strong correlation, okay? Now, like I said, the clock CPGs, just from 300 over, you can predict the age. They are representative of the genome. Now, like many things in biology, what's interesting is actually not those that are within your prediction, but those that lie outside the prediction, basically the outliers. Now, in this case here, you can see this figure here. Again, chronologically, this is people's DNA. And you can see again, this is the chronological age of the subjects and then the epigenetic age in the x-axis. Do not mistake the number 42 and number 43 as ages. Those are data sets, okay? They're just individual points coming from different data sets. Now, if you look at this person, this person here has a chronological age of about 60, and yet his epigenetic age is about 90. Okay, so this poor chap, this is somewhat older epigenetically than this other person, who is actually about 60, 70, about 80, and yet has an epigenetic age of about 50. So these two different sorts of deviation from the mean referred to as age acceleration. It's a terrible word, but it's used and so it sticks. So this would be called positive age acceleration, meaning you are aging faster. Your epigenetics are showing that you are older than your chronological age. And the other one is negative age acceleration, meaning you are younger than your chronological age, as measured by that you are younger, by your epigenetic age is younger than your chronological age. Now, so that's fine. So what does it mean? What do these age accelerations, be it negative or positive, mean? Since this epigenetic clock was published in 2013, many groups have been capitalizing on data sets they already have, and some decided to generate new data sets of people with different conditions. And here, since 2013, and 5 years since then, you can see the kind of diseases and conditions that have been associated with epigenetic age acceleration. You can see the pathologies here, ranging from Alzheimer's, okay, right down to viral infection. And this is just from 2014 to 2019. I stopped collecting the kind of data because there's so much out there. These are pathologies. And not only are pathologies associated with acceleration of epigenetic age, but also health conditions, physical health conditions, mental health conditions, you can see over here, and lifestyle, not necessarily conditions, but lifestyle, environment, and even primary traits, okay, are associated with epigenetic age acceleration. Remember, acceleration can be negative or positive. So I'm just using the word acceleration to encompass deviation from the mean in this case. Right. So in some ways, although the clock was generated or derived purely from chronological age of the samples, somehow it has, in some ways, inadvertently captured some biological aspects associated with chronological age, which is quite exciting. It was something that they did not think they would capture, but it was captured nevertheless by the clocks. Okay. Now, remember, the epigenetic clock was generated using data sets, large data sets from thousands of people. Okay. Now, it is a bit. One has to be careful when you get a certain correlation at population levels with large numbers. It does not necessarily mean that it would be applicable for the individual. Very much like if you can tell the direction of the movement of a gas, a body of gas, you can't possibly say you can tell the movement of every single atom of the gas. Okay, because atoms can be moving in any direction, but on average, they can be moving in one single direction. So likewise with this, so we've got an epigenetic clock that can tell age and everything, but does it work at the individual level because it's been derived from a human population, right? So that was the question when I started dabbling into this area. And I had a good fortune of working with dermatologists, and so we could get healthy skin cells, okay, from patients, from people who present themselves in the clinics with basal cell carcinoma. So you can see over here, you can see this is the basal cell carcinoma, but you can get healthy cells that are beside it, okay, like for example here, the black dot here. So we get it from several patients and we know their chronological age. We take DNA from the healthy cells and then get the epigenetic age using the epigenetic clocks and compare it with the chronological age. And here you can see a very nice correlation of each individual, okay, with between their chronological age and their predicted epigenetic age from there. But at the same time, it also gave us an opportunity to test whether a disease tissue, in this case from the same person, but the cancer itself, okay, what would that epigenetic age of that cancer be? And for each of these individual persons here, their corresponding cancer ages, their cancer cell ages are in red. And you can see the correlation is now nowhere close to their predicted age. So you can see that in some ways, again, that the epigenetic clock, although it was derived using human age, chronological age, somehow it captures the biology that goes awry in, at least in this case, in basal cell carcinomas. So we know the clock works in population levels. We know the clocks work on individuals. And next we ask is, would the clock still work if we took cells out of a person and put it in a dish and culture these cells? Would the clock still be ticking? I mean, I'm sure we can measure the age, as I said, we showed you, but would the age of these cells, would these cells continue to age in culture? In other words, would the clock continue ticking in culture? And they do. So you can see over here, neonatal keratinocytes that were put in culture, and this is population doubling in the x-axis, and the y-axis is their age. And you can see as they grow in culture, they age in culture. You can see this in keratinocytes. It's a kind of skin cells. Fibroblasts is another type of skin cells. Blood vessel cells, endothelial cells, whether they are from children, neonates, or from adults, you can see when you put these cells in culture, the clock they continue to age, and they're picked up by the epigenetic clock. Okay, so this got us really interested because for once we can actually measure age of cells in culture, which means that for once we can actually meaningfully study aging in culture. Now I can tell. Now we know that it works in cells in culture, but when does the clock actually start ticking? I showed you some of the cells there, and I said they are neonatal cells, so they're starting with 0 years old and they age. But does the clock start ticking only when they are at 0? Will it tick earlier? So now, as I mentioned, you can take adult cells, you can measure the age. In this case, a 22-year-old man, you measure the age is 22 years old, and you put that cells in culture, it ages, right? In this case, right up to 80 years old, you can continue, it will continue aging. But what if you took cells prior to those cells becoming an organism, becoming a human being, okay? In this case, embryonic stem cells, which is really, really very, very early on after conception, okay? Would you still be able to measure their age, number one? And number two, then if you were to culture these cells, put these cells in culture, would these embryonic stem cells also age in culture, just like the primary adult cells, okay, that I showed you here? So we took these cells and we measured their age, and their age is prenatal, below 0, which is what you would expect. I did not think it would work that well, but that's what you get. You get cells, embryonic stem cells, measured by this clock age to give you a prenatal age. And if you were to take these cells, these embryonic stem cells, and put them in culture and just keep growing them, passaging them as they become confluent, you just keep moving them, you find that even right up to 100 times of culturing them, passaging them, their age remain largely prenatal age. That means the clocks in these cells do not tick, while that of the adult cells will tick as you put them in culture. Okay. Now you could say, well, it makes a lot of sense, because if embryonic stem cells were to age, then we would all start out with already some degree of aging before even we develop into an embryo. Now you can then ask, okay, so the clock doesn't tick in embryonic stem cells. That's great. But when does it start ticking? Because that was the original question. When does it start ticking? At least we identified the cell type in which it doesn't tick. Now, one way we can ask this is okay, the reason we could use embryonic stem cells is because embryonic stem cells were around at the time, and they are particularly interesting because they can form an entire human body. So even embryonic stem cells in culture can be coaxed to form another cell type. Okay, almost any cell type you wish. If you know how to do it, it will become that particular cell type. And by doing so, we refer to this process as differentiation of embryonic stem cells into another cell type. So we took embryonic stem cells and differentiated it into blood vessel cells, right? Blood cells that coat your blood vessel. And then very early on, we measure the age of these embryonic stem cells, which do not age, the clock doesn't tick. But when you differentiate these cells, very, very early, very quickly at the point of differentiation D over here, you can see the cells immediately start to undergo epigenetic aging. You see the orange, the green, and the blue lines are the different independent experiments where the cells have been differentiated to become blood vessel cells, and they start to increase their epigenetic age, while the parallel undifferentiated cell remains in the same prenatal age, very, very low. So we know that the epigenetic clock starts ticking from the moment of differentiation or specialization of embryonic stem cells into tissue-specific cells. Okay. Now, of course, one of the questions that was put forward very, very early on is like, okay, if we can see these, now we know when this clock doesn't tick, we know when the clock starts ticking, and we know it's very accurate. Can you reset the clock? Okay. One way to reset the clock, of course, you would say is, how do you test it? You can say, if only you could take some adult cells and bring it back to an embryonic state, would you be able to reset the clock? Now, you can't actually get an adult cell to become an embryonic cell per se, but you can do something very, very close to it. And that's what Shinya Yamanaka did. He found that you could introduce 4 factors or 4 genes, okay, which we refer to as Yamanaka factors today, or as O, S, K, and M. If you introduce 4 of these factors into adult cells, any adult cells, you can convert these adult cells back to a state very similar to that of embryonic stem cells. But they are not embryonic stem cells because they did not come from a sperm and an egg. They came from an adult cell in which you introduce OSKM. So we refer to these cells as induced pluripotent stem cells, okay? Because they can do almost everything that an embryonic stem cell can do. So now, when you take this 22-year-old cell, cells from a 22-year-old person, introduce OSKM into them, now these cells have become induced pluripotent stem cells. So what would their age be? Would you be able to reset the clock? And when you measure the age of these cells, indeed, you can. So in a matter of about 2 or 3 weeks, by doing this process, you can change a cell from 22 years old, let's say, down to an embryonic age, because you can convert the cell from a primary cell, adult cell, into a pluripotent cell. So you can reset the clock very, very effectively there. Now, the question, of course, is that then if you were to culture these cells, these induced pluripotent stem cells in culture, okay, just keep growing them, will they now start to age again? And the answer is no, they remain primarily prenatal in age, very much like the embryonic stem cells. So embryonic stem cells or induced pluripotent stem cells, in these cells, the epigenetics do not change, no matter how long you grow them in culture. They're resistant to aging in this. Now, so in summary, what do we learn from here is this: the epigenetic clock was developed based on big data from human cohorts. They're applicable at the individual level. They capture some aspect of biology which goes awry in diseases, many diseases actually. The clock ticks in cells that are removed from the body. The clock does not tick in embryonic stem cells, the clock starts ticking when the embryonic stem cell differentiate or specialize into tissue-specific cells. And importantly, interesting, the clock can be reset when cells are reprogrammed by Yamanaka factors. Okay. Now, when the clock, when in 2013, when Steve Horvath published the development of these epigenetic clocks, at the same time, at the same year, there was this seminal publication by Lopez and colleagues that outline biological features that are associated with aging, okay? And you can see them over here. I won't go through all of them. And of course, now that we understand epigenetics that change with age, the question is, how do they relate to these features or hallmarks of aging? Now, I will not, of course, go through all that with you. We address that, but I will just talk about one aspect of it, which is nutrient sensing. Okay, if you want to know the details about how epigenetic aging is correlated or not to these hallmarks of aging, you can refer to the paper that we published a couple of years ago. Now, I'll just want to take one example, and this is an example related to what Stan was talking about earlier. So the question here is, does epigenetic aging have anything to do with deregulated nutrient sensing, or vice versa, the other way around? Now, as Stan mentioned, rapamycin is a drug that is very, very well used, very well known in the aging field. It has been given to many organisms, and without exception, it extends the lifespan of organisms that have been tested. Okay, and this is a very good example. These are mice, and you can see the population that were treated with rapamycin have an extended lifespan, in this case up to 17% compared to the control population. Now, one aspect of aging and longevity that is often coupled, but it's not necessarily so, is this: if you extend the lifespan of an organism, it is assumed immediately that you have slowed down their aging. Okay, now you might think that makes a lot of sense, and it does make a lot of sense, and it's quite intuitive, but it's not necessarily so, because you can imagine situations where you can extend the lifespan but not slow down aging. Now, rapamycin has been shown to extend the lifespan of organisms, but not necessarily shown to reduce or change the rate at which those organisms age. Now, just to bear that in mind as a question, okay, now I'll show you what happens when you treat cells in culture with rapamycin. When you don't treat them, they will grow in culture in function of days. You can see on your x-axis, on your y-axis is their age, okay? And if you treat them with a controlled drug that doesn't really do very much to them, you can see the same, they age at about the same rate here. But when you treat cells, the same cells in parallel, but when you treat them with rapamycin, then you see that they age slower. The gradient of the treated cells at which they age is significantly slower than that of the control cells. Now, it's important that the rate of aging is slow, but they also extend the lifespan of these cells, because these cells, the control cells, stop dividing over here because they have reached a point where we refer to them as senescent. They can no longer divide, whereas these cells here continue to divide. Okay, so we have extended their lifespan, and they also reduce the rate of the age of aging. Now, when we did this experiment, independently of this, another group, Lou, were looking at available data sets, human genetic data sets, to look for variants in our genes. We know that our genes are not just a single copy, it's a single sequence. There are variations and variants in almost every gene. And they were looking for variants of genes that were associated with increased rate of aging. That means positive accelerated aging. They wanted to find, because when you do that, you can identify proteins or genes that may be important in dictating the rate at which you age, okay? And when they did that, they found one gene that stood out, and that gene is MLST8. And the gene, the gene that encodes the protein that Stan was telling you about in the introduction. And what does MLST8 do? MLST8 binds to mTOR, the complex that Stan referred to earlier. It binds into this pocket and it stimulates, it activates the activity of the mTOR protein. What has that got to do with our observation here with rapamycin? Is rapamycin interacts with another protein in yellow, and this yellow protein actually binds to mTOR as well, just right opposite of that of MLST8. And what does this protein do? It inhibits mTOR activity. That's how rapamycin works, right? It interacts with FKBP12, this yellow protein, and binds to mTOR and inhibit that this complex. So it's gratifying because the epigenetic clock is able to reveal things from human genetic studies and also to be used in the laboratory to track the aging of cells, and both of them converge on the same molecule of mTOR in this case. Now, this is an example of what I mean by the nutrient sensing as one aspect, one hallmark of aging that is associated with epigenetic aging. Okay, that when you perturb nutrient sensing, which is of which mTOR molecule is central, you can perturb the rate of epigenetic aging. Now, back to the question I asked earlier here is that these mice, they live longer, but do they age slower? And actually they do. If you look over here, these are mice that were given this is the control, and this is the rapamycin treated mice. And you can see their age acceleration is certainly smaller than that of the control mice. So using these clocks, you can say that, yes, you can separate the two and ask the question, if you extend the lifespan of an organism by any intervention, are you actually slowing down the rate of aging, or are you not slowing down the rate of aging? And the clocks are now able to tell you one way or the other. Now, that is just an example, like I just stepped out of that to show you the example of nutrient sensing, mTOR, etc. And that is how epigenetic aging, in this case, you can say, is related to nutrient sensing. Like I said, we did for the other hallmarks of aging, and I'm not going to bore you with all that, but just to give you a quick summary and say, these are the hallmarks that are not associated with epigenetic aging, meaning you can perturb cells with you can perturb these features in cells and you don't change the rate of aging, but then these other hallmarks are sensitive. Okay, that means the epigenetics will be changed by these hallmarks. Now, if you look at them, you can separate these hallmarks into two clusters, 1 and 2. Now, these are the ones on the left are those that are related to epigenetic aging. Those on the right are unrelated to epigenetic aging. And when you look at the two, you can see something that stands out here is that nutrient sensing, mitochondrial function, stem cell function, cell communication, and look at the right, genomic instability, telomere attrition, cellular senescence. The ones on the left are things that any cell will need. The cells cannot do without these. The ones on the right, cells can live without this. In fact, cells can live very well without these. So this is more like programs that the cells require. And these are like more akin to wear and tear, okay? And so in some ways, you can say epigenetic clocks, okay, by testing how epigenetic clocks are affected by these different hallmarks, we have split the hallmarks into two, unifying a few of them and putting the other ones aside. And so we can say actually this function of cellular programs, okay, and wear and tear are probably features that are necessary, or not necessary, are features that contribute to aging, not just one or the other aspect of it, okay? Now, so we can say the epigenetic clocks are related to some hallmarks of aging. It unified several hallmarks into one cluster. And at the same time, it divided that cluster into two. One is programs, and the other is more akin to wear and tear. Now, that is not what we set out to do, but that is what we observe through testing the hallmarks and how they relate to this. Now, the last part of what I want to talk to you about is actually very important because it goes beyond the epigenetic clocks. You will see why it is important to leave the clocks behind, as fascinating as they are. But now we got into this by asking first this question: is this epigenetic changes, are these epigenetic changes, and hence the epigenetic clock, are they specific to humans? Now, this is a work that is carried out by Steve Horvath's group. It's a consortium that I was privileged enough to participate in. And what collectively we did was to collect DNA from animals, from mammals, as many species as we can. At the end, they collected up to about 300 over species, DNA samples from 300 over species with known ages. Okay, you need to know how old the animal is chronologically. Collect the species, collect the DNA, and profile them. And just like how we did for how it was done for human samples. And there, again, you will find thousands of cytosins that change with age across all animals, all mammals. Sorry, just keep it to mammals, just across all mammals. And from there, they could build clocks, okay? They could build clocks for individual species. You can have a clock for a whale, an elephant, right down to mice. The fact that you can build clocks using epigenetics of these animals demonstrates to us the universality of epigenetic aging, that it is not something that you see in just one species, see it across, and how well conserved this is evolutionarily at least among mammals. And I think the crowning point about this study that Steve and his group did was to generate a single clock, just one clock that is applicable across the entire mammalian kingdom. So that particular clock up there, the Universal Mammalian clock, one clock can tell the age of a whale or the age of a mouse. I mean, I don't think there's any stronger evidence of how universal this process of epigenetic aging is. Okay. Now, as remarkable as it is, you can't learn very much if you just concentrate only on the clocks. And I've been talking so much about the clocks, and now I'll ask you to leave the clocks alone and remind you that the clocks represent thousands of cytosins. And if you're going to understand epigenetics of aging, you've got to look at the thousands, not just the representatives of the thousands, which are only a few hundred. So if you remove the clock CPGs, cytosins, and look at the thousands, you can ask, what are the cytosins what are the consensus cytosins that change with age across all mammals? And you put that out in a scaffold of the human genome. You can put that out, and you can see this. You see each individual dot here are cytosins on a human chromosome. You can see 1, 2, 3, 4, the numbers there. Those are the numbers of chromosome 1 to chromosome X and Y. And you can see those are the dots. Sorry, the dots represent CPGs in those chromosomes, that become increasingly methylated with age across all species, across 5 different tissues of these species. So not only are they across species, they're also cross tissues. So these are very well conserved cytosins whose methylation increase with age in mammals. And then in blue at the bottom are cytosins that lose methylation, decrease methylation with age. Clearly, you don't need stats to tell you there are more that increase with age than those that reduce with age. Now, so when you look at it like that, most biologists will immediately want to know, is there a sequence specificity to this? Are there sequences in the genome where these age-related cytosins reside? And if you look at the sequence of these age-related cytosins, you will be very disappointed because you'll find that there is no DNA sequence consensus as to where age-related cytosins sit. But that's not actually the end of the story, because remember that DNA doesn't exist just, well, it exists as a sequence, of course, but that's not its only form. That is not its only functional form, because DNA is wrapped up, okay, around histones. And when they're wrapped up around histones, we refer to them as chromatin, and these chromatin constitute our chromosomes. Now, the chromatin may look in this depiction here as just very regular, but that's a bit of a lie, because chromatin, like I said, is when histones, when DNA wrap around histones, are not identical, because histones can be modified. How? They can be modified by these chemical reactions in brackets here. And when you modify these, you add these groups to histones, you actually add what we call histone chromatin marks. Okay, you mark histones differently with these different marks. And chromatin marks, okay, order themselves to a higher order structure. Okay, and those structures constitute what we call chromatin states. Okay, so although depicted as very uniform, chromatin is anything but uniform. There are different states, okay, which are determined by the marks on the histones, okay, which are the proteins like DNA wraparound. And there are so many states. We are still in a process. We, as in the scientific community, are still in a process of honing down to how many states there are. But it's safe to say there are up to about 100 different types or 100 different states, 100 different chromatin states in at least the human genome. Now that you know that there's such a thing as chromatin states, so since age-related cytosins do not correspond to any sequence, we ask, do they reside in any specific chromatin states? Okay, and here I'd just like to introduce 3 different chromatin states that you need to know. Is a bit of a primer before I show you the results, so that it'll be more meaning to it. Is you can have modifications that that bring chromatin together. You can have modifications in blue here that makes a chromatin go a little bit further apart, more loose structure. And this would be a chromatin state that is associated with repression. So the genes next to it or within it cannot be expressed, they're repressed. And here would be, of course, genes close to or at this point are expressed. The expression is permitted. So those are the 2 examples of the hundreds of different states, 100 states you can have. Now, there is one more state, which is a very peculiar state. It has the blue and the red. That means it has marks of histones that are normally associated with repression, and yet it has also at the same time marks that are associated with expression. So you have those kind of in between, and those are called silence or pause states. Okay, and in fact, they are more often referred to as bivalent states. Okay, they are not one or the other; they are bivalent states. So those are 3 examples of the 100 states that you can have. So back to the question, where do age-related cytosine, okay, reside in function of or in context of chromatin state? And back to this figure, if we take a look at the ones that increase methylation with age, the cytosines, the red dots, and what you see at the bottom here, these are the 100 chromatin states. And you can plot a graph and see where the red dots will reside, and you find they are not evenly distributed across all states. You find that they are clustered primarily at these 5 or 6 different regions. And these states are collectively known as bivalent chromatin domains. Just to remind you, they are the ones, the domains, the regions that have the blue and the red modifications, bivalent, okay? Now that is quite curious. Why would age be related to methylation of these domains, methylation of DNA at these domains? To appreciate that, it's important to know how are these bivalent domains made in the first place? How are they generated? You have a protein here called PRC2. You don't need to know more than that for this thing. PRC2 will add modifications to the tails of these histone proteins, in green histone proteins, okay? And they will convert that particular region into a bivalent chromatin region or bivalent promoter, okay? So PRC2 is the complex that does that. It causes a cell to have this bivalent chromatin state. Now, you need to know, to appreciate why this is important for aging, is the bivalent chromatin states are found primarily to be in promoters that control genes that are absolutely essential for the process of embryonic development. Over here, you can see these are different genes. I won't go through that with you because they're very complex, but these genes in different colors, they correlate with the color that you see over here. Okay, the tail, green, and this is where the genes, this particular genes, these genes would need to be expressed. The ones over here in orange are places where these genes need to be expressed. Basically, these genes, they dictate the patterning of the embryo during development. Now, why am I telling you this? Because the expression of these genes are regulated, they need to be regulated very, very carefully for a normal embryonic, for normal embryonic development. And for that to happen, the expression of these genes need to be regulated very, very tightly. And this is where bivalent chromatin regions are found. They are found next to these genes, regulating where and when they can be expressed. If you get that wrong, then there will be abnormality in embryonic development. Okay, it's an extremely intricate process, but highly conserved evolutionarily, and bivalent chromatin domains play an important, pivotal role in determining the time and the position of expression of these genes. Now, we know the importance now of bivalent chromatin domains in development of the embryo. Now, to put it in sequence of events, in embryonic stem cells, that means these are cells that form just very early on after conception, most of the promoters that control the expression of developmental genes are in a bivalent state. They are silent, okay? They don't make very much, if anything, proteins there. During embryonic development, some of these genes need to be expressed. So now the chromatin state changes and become in an active form, and now genes are expressed. But when they are done doing their bit in that particular place, those genes need to be shut down and they are repressed. They are repressed by another modification that will cause the genes now to be repressed. And these genes will never ever be expressed ever again. But life would be very easy if that's the case, but that's not the case actually, because while some genes are truly, truly irreversibly repressed, other genes are actually not repressed, but they are silenced. How are they silenced? Back to this. They're silenced by PRC2 coming in and putting those red marks on them. Okay, so the final products seem to be the same. Both are not expressing their genes, but this is silenced. This is irreversibly repressed. Okay, although the outcomes seem like the same, but the way it's done is different. Now, this is where PRC2 is necessary, not just to establish the bivalent promoters. They're necessary to also maintain this bivalency. You need to maintain the bivalency, and you'll see why. In function of age, as we age, what happens? It is these regions, these bivalent regions that I'm coming back now to, that become the DNA of these regions, not the histones, the yellow bits. The DNA of these regions become increasingly methylated, okay? And this is what the epigenetic clock is measuring, the increased methylation of DNA, okay? And many of these DNA are in bivalent regions. And when this particular DNA is in this region is methylated, what happens? Why does it matter? Because when it's methylated, PRC2 is unable to dock on these sites. And as such, it is unable to put in those red modifications on histones. What does it mean? It means as we age, we have these methylation buildup. PRC2 can't land on dock on this site, and these sites lose bivalency. Okay, because now they only have the blue, for example, or some other colors, but they definitely do not have the red anymore. So you're losing bivalency with age. What is the consequence of that? Remember, they control expression of genes. So if you lose bivalency with age, what is the consequence to gene expression? Well, this is about the last data thing I want to show you. Is I just want to explain to you, this is just for you to understand what is the consequence of losing bivalency. On your y-axis is methylation levels. Okay, you will see a lot of dots in this graph when I remove it, when I remove the white, and each of these dots represent a promoter, a bivalent promoter that is linked to a gene. Okay, if that DNA or promoter is increasingly methylated with age, you will find it above 0 over here. If it is demethylated with age, you will find it here. And of course, it's no surprise to you, I've already told you, most of them become methylated with age. So you will see a lot of dots above this line. On your x-axis, you will see the dots you will see the x-axis is the expression of the genes next to these promoters. If in function of methylation, they increase their expression, you will see those in green. If they further repress the expression, it will be in red. If there is no change to the expression, you will see them as gray. Okay, I hope that's, it's difficult to represent this without a very complicated scheme, but this is what you see. Most of these regions become methylated with age. So that's right, you saw that, and I've been saying it many, many times. But what you see here, these are levels of expression. Each of these are genes that are controlled by this sort of promoter. And you can see, indeed, there are some dots here that are green. That means in function of losing their bivalency, they increase expression, yes. But the vast majority of these dots are not green. They're gray. That means there is no change to the expression. And in fact, quite a few of them have even lower levels of expression than before. So what we have here is silence promoters, which means that when they were in bivalent state, they were silent. In function of age, they become methylated and they become repressed. So they go from silence to repressed in function of age because their DNA becomes methylated at these regions. Now, finally, my last slide to say, why is this so strongly associated with aging? Why is that important? Because a lot of these developmental genes that I told you needed for development of embryo, we are beginning to realize now they are not only important during embryonic development, they are also important when we are important for the organism as adults. Okay, here's just an example of one. HOX, which is one of the developmental genes. It's a classic developmental genes, okay, that specify how your body is laid out. HOX genes in adult skeleton, okay, novel functions beyond embryonic development. This is just an example. I like the second one, the title, HOX service warranty extends to adult bone repair, demonstrating that what we thought of as development-specific genes, now we realize are necessary for maintaining adult tissues and adult health. You can see this in bone repair very, very clearly. If you don't allow developmental genes to be expressed when you need to repair bones, you can't repair your bones. And you can see this, that if you remove PRC, that enzyme that modifies histones, you lose stem cells. And stem cells are the cells, it's a reservoir cells for the different type of cells that your body needs. So you can see that in skin, a stem cell will have to make different type of skin cells. And in order to do that, it needs to it needs to tap into its developmental processes, okay, that are normally used only in development, but we thought initially was only during development. We need to use them even in adulthood, okay? And sorry. And likewise with muscle, etc., etc. So in short, in summary, the process of aging, as we see it now, is embedded in the process of development, in the process of how we come about to be us. It's where the process of aging actually is embedded. And the mechanism that goes awry and lead to aging is the very mechanism that's essential to bring forth life. And this just happens in every single person. We do not know why. We do not know why those regions, the bivalent regions, become increasingly methylated with age, but we know the consequence of that methylation. So I started by saying, what do you think? Do you think the aging is damaged? Do you think aging is programmed? I hope you will. I won't tell you the answer because there isn't one. It's more how you think about this. I hope what I've just shared with you will give you a bit of food for thought as you consider this particular question. And I hope that was clear enough. It's a bit complicated, I know, and it just leaves me to thank the few people that I worked with for over a few years, and they have really, really taught me a lot about the epigenetics of aging. Thank you.